chapters published by leading international publications. So, in addition to his research, Professor Bhaskar, dedicated academic mentor and educator, he has been a regular reviewer of several national journals and serves as an editorial board member in multiple publications. So, he recognizing his outstanding contributions in teaching and research. Guru Asidas Vishwavidyalaya honored him with the Industry Teacher Award in 2020 and 2021 and 2023 also. So, this is a brief introduction of Professor L.D.K.S. Bhaskar, Professor of Biotechnology from Guru Asidas Vishwavidyalaya.
So, okay, thank you very much, Venkatrayalu, for your kind introduction. Before I begin my lecture, I would like to express my sincere gratitude to the organizers for providing this particular platform to interact with you all. And a small laboratory observation made decades ago that sparkled the journey of challenges and recoveries that transformed our understanding of the fetal hemoglobin. Today, I want to share the story of the fetal hemoglobin with you, and the title of my lecture is Unraveling the Role of Fetal Hemoglobin in Sickle Cell Disease, from Gene Regulation to the Curative Approaches. So before understanding the fetal hemoglobin, I would like to give you the small introduction for the what are the different hemoglobins in the different life history stages. So everybody knows that hemoglobin means only one hemoglobin, that is the 2 alpha, 2 beta globin chains are there. So there are actually, we have different hemoglobins, adults and fetal hemoglobins. And this, so am I audible?
Sir, one second. Sir, can you speak now?
Yeah. Am I audible now? Is it clear? My slides are moving.
Can you speak?
Yeah. Am I audible, sir?
1 second, sir. It's not audible. Can now?
Yeah, am I audible now?
Yes, sir.
Okay, my slides also, just check, my slides are moving. Slide transition is happening, sir. Hello? Slide transition is happening or not? Okay, sir. Am I audible now completely? Okay, sir. Okay. Thank you very much. Thank you very much, Dr. Venkatrayalu. Thank you very much for your kind introduction. And before I start, I thank you all for giving me this platform to interact with various delegates that attending this conference. And the title of my lecture is Unraveling the Role of Fetal Hemoglobin in Sickle Cell Anemia, from Gene Regulation to the Curative Approaches. So everybody knows that hemoglobin is a very important molecule, and it is helpful in the transport of oxygen to the various organs, even to the cellular level. So if you see the hemoglobin in the different life history stages of the human development, during the embryonic stage, distinct hemoglobin types are present, which are different from that of the fetal and adult hemoglobin. These hemoglobin variants can be distinguished based on the specific combination of the globin chains that they contain. So for example, HbA, HbA2, HbF, these are classified as the adult and fetal hemoglobins, while Goer 1, Goer 2, Portland 1 and Portland 2. or the embryonic hemoglobins. So the globin chains in these hemoglobins differs across the developmental stages. For example, you take the adult hemoglobin and fetal hemoglobins, 2 alpha, 2 beta chains will be giving the adult hemoglobin, 2 alpha and 2 gamma chains is giving rise to the fetal hemoglobin, 2 alpha and 2 delta is giving rise to the hemoglobin A2. And in addition to that, epsilon and zeta chains also present. They are in different combination, forming the 4 different kinds of hemoglobins, that is Goyer 1, Goyer 2, Portland 1 and Portland 2. So this slide shows you about the ontogeny of different hemoglobins and in the developmental stages, and gene expression varies. Actually, alpha and beta can be seen only after the birth. So alpha and beta, higher level of beta can be seen only after the birth. Before, at the time of the birth, you can just see this dotted line indicates the birth of the individual. At the time of the birth, 90% of the hemoglobin will be the fetal hemoglobin. Before birth also, in the fetal stages also, hemoglobin will be the fetal hemoglobin. And in the embryonic stages, you can find the alpha, beta, and in combination with the zeta and epsilon giving rise to the Goyer 1, Goyer 2, and there is a switch in the embryonic stage to the fetal stage. From the fetal stage to the adult stage, you can see the another switch of the hemoglobins. So this type of hemoglobin variations can be seen till only up to 6 months of age. You can just see the fetal hemoglobin higher level, approximately 90%. Once the baby's age crosses the 6 months to 1 year, you can just see that adult hemoglobin will be dominating. Approximately 90 to 95% will be the adult hemoglobin. Fetal hemoglobin will come down to from 90% it will come down to the 2%, and HbA2 will be less than 4% will be there. So this is the scenario of the hemoglobins in different life stages. That means any individual, adult individual, more than 1 year of age individual, 90% of the hemoglobin is the adult hemoglobin, not the fetal hemoglobin. Fetal hemoglobin will be vanished. So what about the sickle cell anemia? So sickle cell anemia is an inherited blood disorder. Of course, we are not having this particular disease in Andhra Pradesh, because it's Andhra Pradesh also, it is present only in Rajadara or Kondadara and 1 or 2 tribes it is present, but it is entire Andhra Pradesh, it is not there. It is not there even in the tribal populations, it is not there in the caste population, even in Telangana also you cannot find. If you go to the Tamil Nadu, maybe you can just see that in the Nilgiri Hills sickle cell disease is there. That's why many of them doesn't know about this disease and the burden what the family is facing with the kids having this particular disease at the home. So we don't know. But here Chhattisgarh, more than 40 to 50% of the families, they suffer with this particular disease, and the sickle cell anemia is an inherited disease, and this is resulting because of the presence of the mutated hemoglobin, that is the sickle hemoglobin. So here what happens is here CTC is there at the nucleotide level. You can just see that normal hemoglobin, and if CAC is there, that means middle nucleotide at the 6th amino acid position, T to A change will change the amino acid from glutamate to the valine. This glutamate is the highly negatively charged amino acid, whereas this valine is not that much highly negatively charged. It's a near neutral or less negatively charged, we can say that. So the oxygen levels are low in the sickle cell anemia patients. This hemoglobin will polymerize and it will change the shape of the RBCs from the donut shape to the crescent shape, and this type of individuals will be suffering with the problems. So what happens? Actually, this mutation of 6th amino acid will change the glutamate to the valine. So in the normal individual, glutamate will be there, in the sickle individual, valine is there. Once the valine is there and into the adjacent hemoglobins 85, 86, there is a hydrophobic notch, it will go and exactly accommodate there, and one hemoglobin molecule will be attached to the other hemoglobin molecule. So like this, So valine of the one hemoglobin, 85, 87 88 amino acids of the other hemoglobin will attach to each other. So because of this, what happens? A chain-like structure, hemoglobin will be attached, hemoglobin will be polymerized. So X-ray diffraction studies has clearly shown that 7 pairs of this type of polymers are merged together, and a rigid hemoglobin polymer will be formed. This rigid hemoglobin polymer could able to change the shape of the hemoglobin molecule into the crescent shape or the sickle shape. So interaction of different hemoglobin tetramers you see here, and here I just show you that hemoglobin in two different scenarios. Normal hemoglobin, first row is entire thing is the normal hemoglobin, second row is the hemoglobin of the sickle hemoglobin. So that means, so here in the beta globin chain at the 6th amino acid, because of the valine, there will be some change in the valine. Because of the valine, you can just see that there is one hydrophobic end that is generated, and after releasing the oxygen, normal hemoglobin, oxyhemoglobin will be converted to the deoxyhemoglobin A. This is the normal deoxyhemoglobin because of hydrophobic end at 85 to 87. This is inherent notch. Even the sickle hemoglobin you can find, and normal hemoglobin also you can find. Whereas the sickle hemoglobin, additionally you can just see this because of the 6th valine position, you can see the hydrophobic end. So because of this type of structure will allow the polymerization of the hemoglobin, and it changes the shape of the hemoglobin into the sickle shape or crescent shape. So if the hemoglobin is not attaching with each other, you can just see the hemoglobin is freely flowing, or the RBCs will be the donut shaped will be there. They will be easily flowing in even small capillaries also, even in the brain, even the heart tissues, even anywhere in the body, wherever the small capillaries, there also the blood will be easily flowing. Whereas here the small capillaries will be clogged because of this particular deoxyhemoglobin. So this problem will occur only in the oxygen deficiency only. Even as long as oxygen is sufficient is there in the body, there will not be any problem to the individual. So that is about this one. So how we detect, how we identify this particular disease? There are so many methodologies where can be identified. First one is the solubility test, because this RBC, sickle RBC cannot be dissolved easily into the phosphate buffer. Because of this nature, we can easily see that you dissolve in the 5-phosphate buffer, highly concentrated phosphate buffer. If the sample is dissolved and background lines are easily seen, then you can just say that it is a normal individual. And if the background lines are not visible, and that means opaque solution is formed, and transparency is not there in the solution, that indicates the sickle cell is there. Maybe it may be a trait or it may be a disease, but definitely you can see the presence of the sickle cell allele in that. Or at the same time, maybe here even in the slide also, if you make the slide and put some dithionate or the metabisulfate kind of chemical substances, so what happens is this metabisulfate or dithionate will take out the oxygen, and in the oxygen deficient conditions, this hemoglobin will be changed and polymerized, and RBC shape will be changed into the crescent shape. You can just see that all RBCs will not be changed, at least few RBCs will be changed. But these two tests are not that much confirmatory tests. We have to do the, again, electrophoresis. We have to take the hemoglobin and hemolyze the sample, and RBC you have to lyse. That RBC you collect the hemoglobin, and that hemoglobin you can run it on this one. Because of the glutamate, highly negative charge, it will not move that much fast in the electrical field. So the normal hemoglobin will be moving very fast, and sickle hemoglobin will be moving very slow. So one band that is formed with a very slow movement, you can just say that sickle cell anemia, and whereas it is forming two bands, then you can call that as a heterozygous condition, and the very fast band you are getting, that is called as the normal individual. So you can just separate the individuals into. So not only this one, and we are also having the HPLC, hemoglobin HPLC. This mission looks like this. Is not like a conventional HPLC what we use in the chemistry laboratories and biology laboratories. This is the bio-rad HPLC. This is useful for detecting the beta thalassemia or to detect the sickle cell anemia in the laboratories, clinical laboratories. We use this. And here SS disease can be identified, AS disease can be identified, beta thalassemia can be identified. That means hemoglobin fractions can be delineated, and also the quantities of the hemoglobin also can be delineated using this, but quantified, measured with this HPLC instrument. In addition to that, direct sequencing, you do the DNA, isolate it, and you just sequence the individual DNA, and if you identify any mutation, find any mutation in the 6th amino acid, that indicates that person is having the sickle cell anemia. So there are so many methods to do the sickle cell anemia. Who is affected? As I was saying that in the beginning of my lecture, and it is not present in Andhra Pradesh, where it is. So it is present in some parts of the world. Exclusively, it is dominated in the Africa. Africa, you can just see that it is most prevalent in the tropical part of the Africa, and including the countries such as North Ghana, Benin, Central African Republic. In Asia, if you see that, you can just see in some of the populations like Burma, Thailand, Laos, Cambodia, Malaysia, Indonesia, in our India also. Even in our India also it is present in that. So in America, you can find this particular disease in the large populations of African descent. That means there it is present in the African Americans. Americans also it is there, but it is exclusively present in the African Americans. Only black people you can just see that. So in India, where it is? So in India, it is present in only few tribal populations, 1 or 2 tribal populations of Andhra Pradesh, but the frequency is very less. But if you see that 42,000 newborns are affected every year in India. Sickle cell burden in India, it is very much high. And it is 50% of the world cases is present, can be seen in India itself. So the presence of beta S gene in India was first reported by Lehman and Kutbush in the Nilgiri Hills. Nilgiri, that tea plantation people, that Assam people living in the Nilgiri Hills, that can be seen. And today, SCD incidence and prevalence remains particularly high in the central and western regions. Wherever that Karka is there, that Karkataka Rekha is there, that line, Tropic of Cancer. So that line, you can just see that whatever the states that are passing through the Tropic of Cancer, you can just see the high prevalence of sickle cell anemia. So India is estimated to account 50% of the global sickle cell patients. So 50% in Africa it is there, 50% in India is there, other parts it is not much. So the carrier frequency is also ranging very widely. Some populations less than 5%, to some populations more than 45% is there. In Chhattisgarh, some of the communities more than 30 to 40% of the individuals are suffering with this particular disease, having this particular allele. Sorry. And it is a chronic multi-system disorder. So it affects nearly every organ system of the body, with the complications evolving across the lifespan, throughout the lifespan. In slide you can just see that. So these characteristics also, the signs and symptoms also not present uniformly throughout the ages. So at when the young age is there, in the infancy or the childhood, the dactylitis, splenomegaly, and then recurrent infections and early stroke are very common. During the childhood days and adolescence, patients may develop the ischemic stroke, learning difficulties, retinopathy, pulmonary complications, gallstones, avascular necrosis, osteomyelitis, so many complications, various can be seen. Even in the adults, if you see that like long-term complications, chronic complications, like they become prominent. For example, a diabetic, once a person becomes a diabetic, after 10 years, his kidney will be failure. After 20 years, his neuropathy, 10 years neuropathy, 20 years kidney will be failure. After that, retinopathy will come. Like that, chronic complex. Sickle cell also is like that. Sickle cell will come with the birth, but after 6 months, they will see the symptoms. And these symptoms or chronic symptoms can be seen even at the later stages of this one. Proliferative retinopathy, pulmonary hypertension, chronic lung, liver disease, avascular necrosis, arthritis, and also the chronic kidney disease. These are all the things. And priapism is also one of the classic feature in African patients, not in Indian patients. We didn't see that this one. That means these people, erectile penis, prolonged erectile penis will be there in the individuals. So across all ages, sickle cell is characterized by acute episodes such as painful crisis, acute chest syndrome, and stroke. So people mainly suffer with the pain and also fevers. And a stroke and all, you can be seen very, very periodically in the individual. Progressive chronic organ damage leading to the functional decline in the individuals. So what are the basic differences between the normal individual's blood sample or the sickle individual blood samples? So sickle, if you make the smear of the individual blood, normal individual blood and sickle individual blood, normal individual, if you observe under the microscope, you can just see that they are very disc shaped and they are very flexible, and lifespan is approximately 120 years. Whereas sickle cell blood samples, they can live only very short period. So they less than 20 days. If the body is healthy, then 20 days. Even if it's not that much healthy, even less than 20 days only. So these cells become sickle shaped, and also they are very rigid. That's why they will not be dissolved properly in the phosphate buffer that we use as a screening method for this one. And how it is passing to the next generation. So like diabetes and other complications, we cannot identify if the father is having the diabetes, whether son will get the diabetes or not, we don't know. So if the parents are having this particular defective allele, definitely there is a chance of getting 25% of chance of getting this particular disease. That's why we can say that this particular disease in autosomal recessive fashion. It is a genetic disorder. Autosomal recessive fashion it follows. Autosomal recessive means both parents should be obligate carriers. It is mandatory. The person should have the allele, then only it will come. One person is having the allele, other person is not having the allele, it will not come. So it will be possible only if both the parents are having this particular disease. So next, hemoglobin HPLC is a reliable technique used to separate the different forms of hemoglobin, and also it will be helpful for measuring the hemoglobin quantities with high accuracy. And it is particularly useful for detecting the abnormal hemoglobin variants. For example, if you go to the Punjab region, HBD is one more disease is there. If you go to the Bengal region, HBE is one more disease is there. So this type of other variants also, this HPLC instrument can be identified. Almost all the individual hemoglobinopathies such as sickle cell anemia, thalassemia, all the things can be identified. HPLC often shows a higher level of fetal hemoglobin also. So you just see in the pictures, SS beta thalassemia and HBSS individual. I just highlighted the F peak. F means fetal hemoglobin. So individuals with the sickle cell disease, individuals with the beta thalassemia kind of conditions, definitely they will be having this one. So when you are doing the HPLC, if you find the fetal hemoglobin, then you can just say that that person is having the either sickle cell disease or the beta thalassemia disease. Otherwise, normal individuals, our fetal hemoglobin will be less than 2%. If any individual is having more than 2%, definitely that person will be having some blood related disorder. This is the indication. This is one of the diagnostic marker we say that. So as I told that this lecture is concentrating on the hemoglobin, not on the sickle cell much of this one, and hemoglobin F to hemoglobin A. So as I told that embryonic hemoglobins, when the fetus is developing, the embryonic hemoglobins are converted into the fetal hemoglobin. So this fetal hemoglobin after 6 months of the birth, then it is automatically converted into the HbA, that is called adult hemoglobin. And this type of hemoglobin, so this switch, 2 times the hemoglobin switch is happening in the life stages. When from the embryo to the fetus, one switch, and the second switch is happening from the fetus to the adult. So this switch is happening. So what is happening here in the HbA to HbF to HbA switch, we have to understand, because higher level of fetal hemoglobin in an individual is reducing the complications and protecting the sickle cell patients or the beta thalassemia patients from the complications, and complications are very less. They will be absolutely normal individual, like that they can lead their life. So fetal hemoglobin is very important. Higher fetal hemoglobin to maintain in the body, that will be helpful for the sickle cell patients and thalassemia patients. So this switch of the fetal hemoglobin to the adult hemoglobin happens when the locus control region stops interacting with the gamma globin. So generally locus control region interacts with the this one gamma globin. These are the gamma chains. So when it is interacting with the gamma chains, you can just see the expression of the fetal hemoglobin. Once there is the loss of control on the gamma globin gene, this locus control region will be freed. So this is now free, and it will go and act on the beta globin gene, and then the change of shifting from the gamma globin to the beta globin gene is triggered by 2 proteins that are called as BCL11A and ZBTB7A. This is called as LRF. This LRF means lymphoma or leukemia related factor. So ZBTB7A, these are the two important proteins which turn off the hemoglobin gamma and hemoglobin gamma globin gene. I say that hemoglobin gamma or gamma globin gene, both are same. And once the gamma globin is blocked, the beta globin gene can connect with the locus control region, leading to the production of the adult hemoglobin in the individual. That means even after 6 months, if anybody is having the higher level of fetal hemoglobin, then you can just imagine that the person's locus control region is switching to the beta hemoglobin, not switching to the beta hemoglobin. Just see in the next 2 slides, I can just explain you how with a detailed picture how this ZBTB7A and BCL11A, these things will be interacting with the HBG promoter. HBG means it is the gamma gene. So any protein will be having the gene. That's why the HBG is the gamma gene, and this BCL11A or ZBTB7A will be interacting with that, and then you can just see that. So this gamma globin promoter contains 2 critical regulatory regions that are regulated by these 2 proteins. So this ZBTB7A is known as the LRF. LRF means, I already told you, that leukemia or lymphoma related factor. So this LRF will directly binds with the gamma globin promoter and recruits the chromatin modifying complexes. It immediately recruits the chromatin modifying complexes, this leading to the transcriptional silencing of the gamma globin gene. So gamma globin gene is silenced, means gamma globin expression will stop. So then automatically you can see the repression favors the expression of gamma globin repression automatically favors the expression of the beta globin gene. That means adult hemoglobin switch is happening, and it is representing a key event in the normal developmental switch of the fetal hemoglobin to the adult hemoglobin. This happens every individual after the birth. This happens this may happen within 6 months or maybe 8 months or 1 year. It may varies with the different individuals. So, but this definitely happens. If this is not happening, that person will be definitely having the sickle cell anemia or the beta thalassemia. And molecular basis of the hereditary persistence of fetal hemoglobin is the natural point mutations will be there. So here some natural point mutations can be seen in these particular genes, and some of the deletions on the gamma globin promoter gene can disrupt this particular repressor binding sites, thereby preventing the recruitment of transcriptional repressor. So it will completely fail to recruit the transcriptional repressors. In contrast, large deletions spanning the beta globin cluster eliminate the delta beta competition and reposition of the locus control region. So when there is a competition between the so who has to take this LCR region, whether the gamma globin or the beta globin? There is always gamma and beta competition will be there. Once this competition is reduced, further promoting the gamma globin expression, and as a result of this, individuals with hereditary persistence of fetal hemoglobin maintains elevated higher level of fetal hemoglobin. Generally it is 2%. So sometimes individual 30 to 35% also can be seen in the individual, higher in the adulthood. So whenever you find that, for example, you go to the laboratory and ask for the hemoglobin HPLC, if the hemoglobin HPLC is higher, definitely some blood related disorder can be linked to the particular higher level of fetal hemoglobin, which can be ameliorate the severity. So why this is the nature of this one. Nature always try to offer some protection. So hemoglobin, higher level of body will be protecting from the disease by keeping the higher level of fetal hemoglobin in the body. So severity of hemoglobinopathies will be reduced, sickle cell complications will be reduced, or the beta thalassemia complications will be reduced. So this is because of the higher fetal hemoglobin level. So that is main thing. So what happens, this fetal hemoglobin? Fetal hemoglobin, really is it helpful? Why it is helpful means, so hereditary persistence of fetal hemoglobin, that means after 6 months or 1 year also it is having the life, means it is not associated with the clinical symptoms. People has identified that. So whoever is having the higher fetal hemoglobin levels, they are protected from the disease complications. So their fevers will be very less, their other hospitalizations will be less, and the blood transfusions will be less. And like that, you can just see you are protected from the sickle cell disease complications and avascular necrosis, pain, severe pain will get. So they cannot tolerate, they cannot go to school, they cannot play with the other kids, they will not take the proper food. So this type of complications will be there. This elevated HbF in adults has inspired the exploration of the HbF reactivation as a therapeutic strategy. So now started thinking, scientists or researchers started thinking how to increase the fetal hemoglobin levels in the individuals. So because this fetal hemoglobin is not attaching with the normal hemoglobin as well as the sickle hemoglobin. So if you are keeping the more amount of fetal hemoglobin in the body, what happens? So polymerization will be reduced. So this fetal hemoglobin will not be attached to the normal hemoglobin, and this will not be attached to the sickle hemoglobin also. That's why having the higher level of fetal hemoglobin, that means hereditary persistence of fetal hemoglobin, is one of the important aspect for the therapeutic this one. So that is this one. So whether this fetal hemoglobin is distributed in uniformly in all the cells, all red blood cells are having the uniform amount of fetal hemoglobin. So studies have shown that some people has done some smear analysis and some people has done flow cytometric analysis, and these studies has very clearly shown residual fetal hemoglobin is not evenly distributed among the blood cells, but it is concentrated in a subset of F cell, subset of population, subset of RBCs. Those are called as the F cells, where it constitutes approximately 20% of the hemoglobin. So hemoglobin 20% is there, more than that it is there, that is called as the F cell. So not all the RBCs in the body, it is having the fetal hemoglobin in the animal. So whereas it constitutes 20% of the total hemoglobin, and this uneven distribution can be visualized by staining this. Make the smear and stain with erythrosin B. Erythrosin B will be staining this particular fetal hemoglobin in the on the slide. So this highlights that HbF rich cells can be seen on the smear. At the same time, flow cytometry analysis also provides the broader view for the HbF distribution, and across all cell populations you can just see understanding this localization of HbF in the F cells is very important for studying the protective role of its conditions like sickle cell anemia or the beta thalassemia. That means higher level of fetal hemoglobin, higher level of or increased concentrations of fetal hemoglobin in certain cells, at least 20% of the cell population is there also, that will be preventing the or supporting the body from the preventing the complications, or it will support the complications of this one. So okay, so HbF reduces the symptom. This is a clear observation in 1948. So this is before 1952 our Indian sickle cell patient was identified. In 1948 there is a simple observation in the laboratory. So sickle cell baby is not having the sickle hemoglobin. That means he is having the maximum fetal hemoglobin. Everybody will be, you see this particular slide, one day you can just say 77%, and 5 days you can just 76%, 3 weeks 70%, 6 to 9 weeks you can just say 50%, by the 6 months 4.7%. So it has to come down to this one. Slowly it will be reducing and the adult hemoglobin will be increasing along with the time. So it may sometimes some babies may be having even up to 1 year also. Then we generally suggest that don't go for the any treatment till 1 year. So that means baby is protected because higher amount of fetal hemoglobin is offering already protection in the individual. Then if person is a sickle means definitely will be having 10 to 15 or 20% of the hemoglobin as the fetal hemoglobin. And the newborn babies doesn't manifest the sickle cell complications until they reach 6 months to 1 year. So it varies. That's why. So this protection in the infants is due to the HbF. So why they are not having the complications means this is only because of the higher amount of the fetal hemoglobin in their RBCs. Asymptomatic patients, some people actually, we used to go for the screening in the population or sometimes in the school boys to screen in the chestis because it's very rampant in this particular area. When we identify screening the students, we also do the teachers. The teacher says that I am healthy sir, I never got a fever, I never experienced any pain in my life. But when we screened that person becomes HbSS. That means some people are asymptomatic. That's why this prenatal diagnosis after doing also we are not suggesting that going for the termination of the baby. So asymptomatic patients also, because fetal hemoglobin is already offering some protection to the individuals. So what happens, as I already told you that sickle hemoglobin higher fetal hemoglobin, why it is not happening attaching with the sickle hemoglobin or with the adult hemoglobin is at 85, 87 there is a hydrophobic end that is generated. There actually at the molecular level, if you see that sickle hemoglobin polymerization driven by the abnormal interactions. Abnormal interactions means on one hemoglobin 85, 88 amino acids are having one thing and beta 6 valine is there. They bind with this one. Hydrophobic patch will be formed and these residues will be very adjacent to that. That's why they come and attach to each other. So in contrast to the gamma globin gene at amino acid position, they have the glutamine instead of threonine. So in normal hemoglobin, it is threonine at 87 amino acid, and whereas in the sickle fetal hemoglobin, this is glutamine. That's why they are not interacting with it. They are not attaching with any of the sickle hemoglobin or even with the normal hemoglobin in the individual. So this subtle difference, this one single nucleotide variation. So once one amino acid change makes that weakness in the hydrophobic interactions and reducing the tendency of HbF containing tetramers and polymerization itself. So kinetic equilibrium studies also further confirms the structural expansion or explanation or otherwise highlighting the this one fetal hemoglobin are protecting the individuals from the anti-sickling or the polymerization of the hemoglobin. So here a small study that has identified, that has quantified the fetal hemoglobin level in different individuals, and here that three different color codings, you can just see that HbAA, that is the pink color, and light green color is HbAS, and also HbSS, the light pale blue color, you can just see. You can see the fetal hemoglobin very high in the individuals of the sickle, this one heterozygous condition, it is very less, and with the age increases you can just see this type of thing. And this concentrations, patients with the sickle cell disease have higher concentration of the fetal hemoglobin, unaffected adults, and also largely due to the longer survival of the F cells. So longer survival of the F cells can be seen only in the sickle cell individuals, and the levels of HbF also vary between the individuals, and genetic factors account approximately 60 to 90%. So many of the traits will not be explained by the only genetics. You cannot say that only one particular gene can make the HbLC or the fetal hemoglobin levels higher or lesser. So only genetics will be here useful only for explaining only 50 to 60% or 90% sometimes variations only. Not is very difficult. That much variation will be there in the individuals. So if you see that the molecular level analysis of deletions, so some of the deletions also occurring, actually naturally occurring phenotypes. So naturally occurring phenotypes means take that sometimes you can take that beta thalassemia, sometimes sickle cell anemia, sometimes a condition without any complications, you can just see that hereditary persistence of fetal hemoglobin. Sometimes you can just see the Corfu deletions. So this type of deletions will be, some may be large deletions, some may be small deletions. Beta, gamma and delta globin genes also you can find this particular deletions which increases the fetal hemoglobin production. For example, you see this delta beta thalassemia leads to the modest increase in the hemoglobin, whereas the hereditary persistence will have a marked increase in the fetal hemoglobin. Whereas the Corfu, if it is a heterozygous deletion, if the deletion is there only on one chromosome, minimal change in the fetal hemoglobin. If it is a heterozygous, that means homozygous condition, that means both the chromosomes it is having the deletion, then that will be having the 100% elevation in the fetal hemoglobin levels. So this is about that means naturally occurring phenotypes also have some fetal hemoglobin level. These were also helped us to identify the mechanisms of having this particular higher level of fetal hemoglobin in the individuals. And you can just see this. So linkage scans identified approximately 4 different loci. That one loci is identified in 6p23 region, one is 8q region and xp23 region. And fine mapping of these genes also identified the genes that are responsible for the determination of the fetal hemoglobin level in the individual. So fine mapping of 6p23 revealed that intergenic sequence between the HbS1L and MYB region, and in this region having so many erythroid transcription factors. So transcription factors that are favoring the erythropoiesis have been identified, and reduced MYB expression also increases the fetal hemoglobin level in the individuals. Okay. Genome-wide association studies also, because nowadays we are not doing the candidate gene approach or one gene approach, those things are all as long. Now we are doing the genome-wide association studies. Genome-wide association studies using the healthy Europeans, beta thalassemia patients, African SCD patients, even Asian HbE beta thalassemia populations. They have identified few loci that are determining the fetal hemoglobin levels. Here the BCL11A which is present on the 2pq61, you see this higher p-value. These are all dots are called as the p-values. So higher p-values we got it in this family set, the BCL11A gene only, that is present on the 2p16.1 is emerged as a major globin repressor gene, which the variations increases with the hemoglobin levels. So nearby other chromosome, you can just see the chromosome 11 OR. So chromosome 11 olfactory receptor genes, that is called as OR51B5 and OR51B6. These have been found as a linkage disequilibrium because these are all actually surrogates for this. They are tightly linked with this particular BCL11A, OR51B5 and B6 are tightly, they are in linkage disequilibrium because non-randomness will be there. So because BCL11A, one particular region is having the T mutation, here also will be the T mutation. That is called as a linkage disequilibrium, tight linkage disequilibrium. Thus, the functional effect of fetal hemoglobin, if you want to understand, BCL11A you can study. If you want to use that as a marker, maybe even OR51B5 or OR51B6 can be used because of its strong linkage disequilibrium with the other loci that are determining the fetal hemoglobin. As we are talking the BCL11A, what is this BCL11A? BCL11A is a zinc finger transcription factor. It acts as a regulatory factor for the expression of the fetal hemoglobin levels. So fetal hemoglobin, that means gamma globin gene has to be expressed, means this BCL11 should not be there. So BCL11 always inhibiting the gamma globin expression. So to increase the gamma globin expression, we have to remove the effect of the BCL11A. So in humans, higher level of HbF is associated with the lower level of BCL11 expression, and BCL sometimes what happens in the even animal studies, so BCL11 null mutants mice also exhibited the fetal hemoglobin levels. So if BCL11 is completely removed or BCL11 is suppressed also, you can just see the increased levels of fetal hemoglobin in the individuals. So this demonstrating its critical role in the gamma globin silence because it is having something with in the future. That's why we are, for example, any genome editing technology if you want to use, where we have to touch, so we have to remove this particular BCL11A. So then if you remove the BCL11A effect, automatically gamma globin expression will be increased. Gamma globin expression is increased, automatically what happens? Fetal hemoglobin level will increase. Fetal hemoglobin level increases, complications will be reduced. Complications reduced, the quality of life improves in the sickle cell patients. So this is what that much link is there in between the each thing. So BCL11 is a very important thing. So BCL11 is embryonic and fetal globin gene silencing also will be, you should understand. So MYB and KLF1 do not directly bind with the beta globin genes. So here MYB and KLF genes are there. These two things are directly interacting with the beta globin genes, and MYB increases the expression of the BCL11A. So MYB increases, it acts on the BCL11A and BCL11A expression will be improved, and the major globin repressor, so it silences the globin gene, and globin gene silences, automatically fetal hemoglobin will be shut down, synthesis will be shut down. Like that only TR2 and TR4, this is known as a DREAD complex. So this DREAD complex on the other hand bind directly to the gamma globin promoters and recruit the repressors to help. So once it is recruiting the repressor, what happens? Its synthesis will be reduced, and KLF bind to the beta globin promoter region and boosts the adult hemoglobin, that is called as HbA, normal hemoglobin production, and it also activates the KLF activates the BCL11A. So MYB activates the BCL11A, and also KLF1 also activates the BCL11A. Together, this MYB1 and KLF1 coordinate as a switch for the fetal hemoglobin to the adult hemoglobin. This actually really it happens in every individual. At the age of 6 months, maybe you can just see that ensuring the fetal hemoglobin is silenced and beta hemoglobin is turned up. But whereas the sickle cell patients and beta thalassemia patients with the hereditary persistence of fetal hemoglobin patients, you can see this particular two genes coordinate as a switch, and they will be prevented if you suppress these two things also automatically. So if you suppress the MYB, BCL11A can be suppressed. If you suppress the KLF1, BCL11A can be suppressed, or otherwise maybe BCL11 itself directly you can target. So this kind of things can be done in the laboratory. So other target sequencing, actually sometime what we do is we do some of the candidate gene approaches and all we're using. So hemoglobin switching genes, regulatory networking involving the transcription factors, co-regulator factors, or even sometimes chromatin remodelers. These are also studied by this one. The core regulators such as GATA1, TAL1 and OCT1, TR2, TR4, and these interact with the key factors like KLF1, DNMT1, BLF, and these will be repressing the beta globin chain. So this GATA1 and BCL11A act as a central nodal for the and identified as a regulators for the fetal hemoglobin. So mutations in sometimes KLF1 or BCL11A1 or the DNMT1 alter the gamma globin regulation. So that's why targeting these genes also will be helpful for increasing the fetal hemoglobin in the individuals of sickle cell. So like that only this recent emerging trend of microRNA gene expression. MicroRNAs are the small nucleotides that are present in the cell, and this is because of their base pairing with very specific base pairing specifically is limited because it's a very short nucleotides of 6 to 8 nucleotides, and they will be finding the binding site for the many RNAs. What happens is this small microRNA will go and bind to the 3' UTR region of the mRNAs. Once it binds to the mRNAs, it may block the mRNA expression, or otherwise it may degrade sometimes mRNA expression. So one particular miRNA can potentially interact with the several of hundreds microRNAs in the individual. That's why if you can target the microRNAs also, you can do this. So several microRNAs involved in the regulation of the hemoglobin expression has been identified. And LET family means so many LET microRNAs are there. They are developmentally regulated during the fetal hemoglobin to the adult hemoglobin transition. That's why some of the identified, this one MIR486-3P, this directly binds to the BCL11A mRNA and reducing the expression and thereby promoting the gamma globin synthesis and promoting the gamma globin expression. And in contrast, inhibitors of MIR26B or MIR151-3 also leading to the decrease in the beta globin or gamma globin expression, highlighting the possible role in this one. If you can manipulate with this microRNA, also you can increase or decrease the fetal hemoglobin level in the patients. And third slide, I want to show in this slide what are the different microRNAs that are expressed, are highly expressed, or less expressed, that means upregulated or downregulated in the normal and sickle cell patients. You said they see this, and when compared with the normal cells, sickle cell individual, sickle cell blood is having some of the genes that are upregulated, some of the genes are there. These alterations are affecting the erythropoiesis, and these may affect the oxidative stress response, and also sometimes they may involve in the gamma globin regulation also. So this upregulated microRNAs includes LET7I and MIR26B, MIR29A, MIR140, MIR142-5V, or MIR144 and MIR451. So the downregulated are LET7 family. Other than LET7I is upregulated. All other LET7 family members are downregulated in this. MIR141, MIR181 and MIR320 also downregulated in the sickle cell anemia patient. So if you can target these particular miRNAs, so that will also help us to alter the fetal hemoglobin levels in this one. Now therapeutics. Now we understand how the fetal hemoglobin is regulated in the body. We have seen different mechanisms, how microRNAs are doing, how some candidate genes were there, how some of the genome-wide association studies have identified locus that are very specifically KLF1, MYB, BCL11A. These are the very important genes. So now we have to use these genes to make the therapeutics. So now people are talking about the gene editing is the next future of the therapies. So therapeutic induction of the fetal hemoglobin. So any chemical that used to induce the fetal hemoglobin in the body can be used as this one. So that's why they identified the hydroxyurea. So to keep the fetal hemoglobin level higher, so they use the hydroxyurea. Hydroxyurea, this is also known as hydroxycarbamide. Hydroxycarbamide inhibits the ribonucleotide reductase and depleting the deoxyribonucleotides, causing the S phase arrest. So cells will be arrested at the synthetic phase. So once the cell phase, this particular arrest of the cell phase, cell cycle at the S phase will cause the stress on the erythropoiesis of the bone marrow signaling and promotes the gamma globin expression, leading to the increased fetal hemoglobin. This one. That's why in 1980s the FDA has approved the hydroxyurea for the treatment of the sickle cell anemia. So till 2-3 years before, so hydroxyurea is the one and only medication for the sickle cell anemia. But this also is not that much easy chemical to operate. You cannot take on counter. You cannot take like diabetes medicines. What we take, we cannot take pain medicine, paracetamol medicine. We cannot take because it's highly risky because it is directly interacting with the ribonucleotide reductase, and also it is arresting the cells at the cell cycles, S phase of the cell cycle. So that's why you cannot take continuously this medicine. You have to monitor the cells, blood cells. CBC you have to do continuously, complete blood count. You have to see, monitor the cells. Sometimes what happens, it reduces the number of the white blood cells, and once white blood cells is reduced in the body, automatically leads to the complication. So this therapeutic induction only through in the doctor will prescribe only by seeing his fetal hemoglobin level as well as his complete blood count before continuing this particular hydroxyurea for longer time. Otherwise, for few times on off they do it. For 3 months they give, again they stop for 3 months, again they increase, again 3 months, again they will increase. So they monitor the cells and give the fetal hemoglobin to the patients. And next approach is chromatin dynamics. So here generally we talk about the methylation. And the methylation, if you talk, the chromatin dynamics will come into picture. Chromatin dynamics and methylation are linked, and HbF switch is always accompanied by the epigenetic changes. Epigenetic change means there will not be any change at the sequence level, but you can just see that acetylation, methylation or simulation are kind of different epigenetic changes will be there. These epigenetic changes will also be involved in this one. So in adults, gamma globin repression is linked to the increased cysteine methylation. So when the switch of the gamma globin to the beta globin, you can just see the increased cysteine methylation, and also lack of active histone modification can be seen in the individual. So the histone deacetylase inhibitors and such as butyrates can be used for this one, can restore the fetal like histone like acetylation in the gamma globin surroundings. That's why locus control region will come and easily approach that one, and then automatically its expression can be maintained. So DNA methylation inhibitors like HDAC inhibitors can have a diverse roles. They may modulate the chromatin accessibility. They may influence the global gene expression and regulate the cell cycle repression and also apoptosis. So this kind of HDAC1 and HDAC could be used to adjust the fetal hemoglobin in the individual. HDAC inhibitors, now it is a trending medicine. It is used for the cancer and used for the apoptosis, autophagy, DNA repair and stem cells. So many places it is being used because the epigenetics is the current trending therapeutic approach for this one. So here BCL11 enhancer. Now we can manipulate the BCL because BCL is one of the primary factor. So in the presence of BCL, fetal hemoglobin is not happening. So somehow you delete the effect of the BCL11A to get the effect of increase the fetal hemoglobin level. What happens? A general phenomenon. So erythroid enhancers in the beta BCL11A increases the beta BCL11A protein. So presence of BCL11A protein, higher level of BCL11A protein will inhibit this particular gamma globin genes, and then lower expression of lower expression of fetal hemoglobin can be seen in the individual. So whereas if you remove this particular BCL11A expression, then what happens is you can just see the presence of, here you see this in this natural mutation. If any mutations are there in the BCL11A, automatically the amount of BCL11A will be reduced, and automatically you can just see the increased amount of fetal hemoglobin levels can be seen. So in case if you can completely delete this particular gene, then automatically you can increase the BCL, automatically you can see the increased concentration of the fetal hemoglobin level. I'm just coming to the end of this one. I'm going to another 4 slides remaining. So high level of fetal hemoglobin can be maintained by removing the effect of this one, by increasing or by manipulating or by reducing the expression by use either fetal hemoglobin or use the CRISPR kind of genome editing technologies to remove this particular thing. So if in case, for example, if you are doing the genome editing technology, it's not that much easy, but it is having its own potential benefits as well as potential limitations or risks also there. But whatever it is, but as an academic lecture, I just talk about this one. It's not a clinical translatable thing in that stage. So collect the blood cells from the person with the sickle cell anemia, introduce the sequence specific nucleotide to disrupt the BCL11A. Maybe you can use some of the microRNA that can disrupt the BCL11A, or otherwise you can use some of the CRISPR kind of technologies like CRISPR-Cas9 or TALENs or zinc finger nucleases, these things, because it's a zinc finger protein. So zinc finger nucleases can be introduced into the cell, and you can just demolish or you can reduce you can disrupt the particular BCL11A enhancer, and these cells, after disrupting the BCL11A, these cells will be reinfused into the bloodstream. You can just again put into the bone marrow, and so that what happens is automatically increases the fetal hemoglobin level. This approach is currently being evaluated by the clinical trials, where the CRISPR-Cas9 edited hematopoietic stem cells have been demonstrated the sustained increase in the fetal hemoglobin levels and marked reduction and even elimination of the sickle cell anemia crisis. So crisis means once they get to separate some conditions, for example, if very severe cold condition or severe hot condition, whenever the body dehydrates, these people will be getting the continuously they get the pain. So this pain they cannot tolerate that particular, they will be crying continuously. So and representing the major step, and this kind of this one complications, reduced complication can be seen. So this is one of the now it is yielding some promising results, and it has to maybe another 10-15 years maybe we can see because it is having so many potential benefits and also limitations. You see this any genome editing technology, if you are talking, so genome editing offers a promising and precise and potentially permanent modifications in the genome. That's why we use the genome editing technologies. So in the context of sickle cell disease, the loss of BCL11A expression increases fetal level. Not all, I'm not talking about only you do the BCL11A, you do the KLF1, you do the MYB, any gene you take it, but BCL11A is one of the enhancer, repressor. That's why if you can remove this particular thing, automatically you can just find this particular increase. So loss of BCL expression increases the fetal hemoglobin, and modification is a permanent and providing the survival advantages to the edited cells. So if you edit it, then automatically it can outcompete the unmodified cells also. However, the potential risks we cannot ignore. So potential risks include unintended genome modifications and off-target tissues. Your main is to repair the blood cell. Maybe it may repair and go and repair something and modify something, and that may lead to some teratomas and some cancers or something. So that type of things also we have to see. So need for the preparative therapy for the stem cell transplantation, immunosuppression, also is needed, because anything you give to the body. So for example, any cells you put it in the body, it will act as an antigen. So that's why you have to put that immunosuppression. So immunosuppression, any of the organ transplantation, immunosuppression is essential, and possible organ complications such as heart, liver and lung failure will be there. Sometimes it may lead to the teratomas as a cancer kind of thing. So in conclusion, this is my last slide and my conclusion. The recent advances in the gene therapy offer the multiple strategies to address the sickle cell disease, including the direct correction of the sickle cell mutation, induction of the fetal hemoglobin, and addition of the functional globin genes by targeting the key regulators such as BCL11A or KLF1 or maybe MYB gene, and leveraging the innovative genome engineering or vector based approaches. These therapies hold a promise of transforming the disease management and moving closer to the potential cure. So with this, I would like to thank the organizer for giving me the opportunity. These are some of my publications, a good number of publications, 30-40 publications we have done in the sickle cell patients of Chhattisgarh region, and these are some of my publications. If you have any doubts or any suggestions or something in my presentation, I welcome you all to give some comments or suggestions on my talk.
Any doubts? Any questions? So this is my daily data. Sir, this is Dr. L.V. Reddy, sir.
Yes, sir. Yes, sir.
Good morning, sir.
Good morning, sir. Good morning.
You have presented very descriptive lecture on sickle cell anemia,
Thank you.
sickle cell anemia, from the scratch point of definition and conventional methods to detection to the very advanced methods like sickle cell anemia, HbF detection, and also molecular methods, electrophoresis methods, very descriptively. It is very much useful to the students.
Thank you.
And we also touch upon therapies to cure the sickle cell anemia, which are very interesting, sir. And you were finally shown the whatever you have done in published papers, which are very nice. Congratulations, sir.
Thank you very much.
Thank you for thank you very much.
Yeah, thank you. So shall I quit now, sir?
This is Chandramani.
Yes, sir.
We can express our gratitude for accepting this invitation and giving this wonderful Sir, now we can start, sir.
Yes, I can leave now, sir. Thank you.
Yeah.
Yeah, thank you. Thank you very much, sir.
So,
బాగా వచ్చిందా సాటి ప్రజెంటేషన్ బాగా జరిగిందా విన్నావా ఎంత అయింది టైము పోలేదు నువ్వు ఎల్లుండి కూడా సరే ఇది లైవ్లో పెట్టి మాట్లాడుతున్నాను నీతోటి
Lupa.